Um, so thank you very much for the opportunity to speak today about our recent work exploring how specific chromatin topologies in specialized cells are involved in gene regulation and gene expression. And this is particularly important and interesting because in complex neurological diseases, often these are polygenic diseases with complex etiology that's unknown, and the disease-associated variants are usually found in non-coding regions, or very often found in non-coding regions. And that suggests that there's a very important function for these regions, especially because we know that enhancers are very cell type specific, and that suggests that the contacts between enhancers and genes are also cell type and cell state specific. And so we really would like to be able to connect then how the specialization of topologies in these different cells may be related to genome sequence variability towards understanding gene expression at the level of transcription, mRNA, and protein modifications. So the question we really wanted to ask with the study here was, how specialized are 3D chromatin structures in the brain, and how do they regulate gene expression and relate back to these potential disease mechanisms? And so to do this, we extended the GAM technology and developed Immunogam. GAM was developed in our lab, in Anna Pumpel's lab, as a tool to study 3D chromatin contact data in very specific cell types. So the aim here was to map chromatin topology in specific cells in situ in the brain. And we took 3 cell types with specialized functions: dopaminergic neurons from the midbrain, which are involved in reward and addiction; pyramidal glutamate neurons from the hippocampus, involved in memory and learning; and oligodendrocytes from the somatosensory cortex, involved in myelination as well as in neuron-glia communication. And to select these cells, we fixed the tissue very highly, extracted the regions, and then we cryosectioned tissue blocks into very thin slices, which we could then label with cell type markers for the cells of interest. For example, in the DNs, we used a protein marker, and in the PGNs, we used a marker to visualize the tissue morphology, to see the pyramidal cell layer there, or we even used a lineage marker in the oligodendrocytes for the GFP. We could then cut out individual cells, individual nuclear slices to extract the DNA and sequence it, and then build chromatin contact maps that are representative of the true physical distance between genomic loci. The goal here really was to then integrate these chromatin maps with cell type specific gene expression programs and chromatin accessibility. So we leveraged published data when it was available, or produced our own data when it wasn't. And then we compared these chromatin contact maps between the different cell types in the brain, but also to embryonic stem cells. So here's just one example of the clustered pcbh locus, which has 3 clusters of cell adhesion genes. This is in embryonic stem cells. Here you see the contact map, and then we need the contact density, which we calculated using insulation score method at multiple distances. And you see that the region's not expressed in ES cells, or at least the alpha and beta clusters are not. The gamma clusters has a little bit of expression, and it's not really open. But then in the brain cell types, so we have the oligodendrocytes, the PGNs, and the dopamine neurons. Now you see that there are contacts more strongly happening between this alpha locus and the beta and gamma clusters, and these are very strongly happening also in the neurons. We also see some very strong cell type specificity. For example, each one of these clusters has strong contacts with the downstream gene FGF1 in the oligodendrocytes, which is involved in myelination. Yeah, I think it's important to note that each of these maps comes from a single mouse. So we can get chromatin maps from single mice, and that allows us to compare between mice. So here is a replicate mice for the two neuronal cell types, and these maps are quite similar. One of the most interesting findings, and this is where I'm going to spend most of my talk today, is we found very large loss of contacts, decondensation, what we call melting, uh, at long, highly expressed genes. So these are genes that are longer than 400 kb. They're often very highly expressed in neurons and have very complicated splicing. And they're very important for neuronal processes. For example, here we have BRIC2, which is a glutamate receptor that's disrupted in epilepsy and in cognitive disability. And if you take a look at the ESL map down where my arrow is, you can see the contacts are quite strong within the gene, but also in the surrounding region, even though the gene is not expressed or it's not open. However, then when the gene becomes expressed in the glutamate neurons, now you see that it's expressed and it's quite open, you actually lose contacts within the gene region. And you can see that quite clearly in the contact density maps, especially at the TSS and the TES. Another example here in the dopamine neurons is DSPAM, which is a long gene involved in cell adhesion and synaptic connections. It's associated with Down syndrome. And here again, in the ES cells, you have strong contacts without any expression or accessibility. But then when the gene is expressed, you lose contacts. And that's, again, very clearly seen in the contact maps. So we have some of these really striking examples, but really we wanted to find these events happening genome-wide and in an unbiased manner. So we developed a pipeline, which we call Meltron, to do this. And we essentially take regions of interest. In this case, it was genes that were longer than 400 kb. And then we calculate the insulation score maps in the gene coding region for the cell type of interest and compare it to a reference cell type. In this case, we always compare to the ES cells. We can then plot cumulative probability maps, probability plots, and then calculate the difference between those two cell types, which we then apply a statistical test and obtain a p-value, which we convert to what we call a melting score. So when we applied the Meltron pipeline to our 3 brain cell types and compared them to ES cells, it was very clear that very many of these long genes, 100 to 150 of the 480 genes that we tested, contain these melting events. And we considered any gene that had a melting score. You can see that on the x-axis of these graphs here. That was any gene that had a melting score greater than 5 had a melting region, or was what we call the melting gene. And quite interestingly, when we compared it to the transcription on the gene, and that's on the y-axis here, we found that genes that were melting had higher transcription, significantly higher transcription, and that was especially true in the PTNs and in the DNs as well. We also saw the same pattern when we looked at chromatin accessibility. Genes that had these large decondensation events had higher chromatin accessibility in all 3 cell types, most notably in the 2 neuronal cell types. So what are the mechanisms that are leading to this type of decondensation, or what does the decondensation mean? And to delve a bit further into this, we took a few example genes and looked in more detail. So here we have Mirexin-3, which melts in the DNs. You see this, the melting region in the second half of the gene, where the gene is quite highly expressed. It's in the A compartment in the DNs, whereas it's in the B compartment in the ES cells, and you have the strong contacts without expression. We also looked at published LAD and NAD data for ES cells and found that it's associated with both lamina and nucleolus. And we wanted to understand then how does this pattern relate to how the region might be folding. So we performed polymer simulations using PRISMR with Mary Nicodemu's lab. And Here's some examples of the polymers for the Orexin 3 region. The green domain here is the melting region, and in DNs you can see it's quite elongated, whereas in the ESLs it's quite folded up. We can measure that unfolding, that decondensation, using a gyration radius, and so we measured that for the ensemble of polymers. We had 450 polymers for each cell type. The arrows here are indicating the polymers we're looking at that are moving right now. And we found that the gyration radii are much, much higher in the DNs, indicating they're much more extended. In fact, the upper quartile has higher gyration radii than all of the ES cells. So this was really invaluable to help us understand how the folding might be happening, but we also wanted to understand how it related to the single cell population and how the gene might be localized in each cell. And so we next looked at another example, RBFOX1, which is involved in alternative splicing, and mutations in the gene lead to epilepsy and autism. And here we had a melting region identified at the TSS for the PGNs. Again, it's in the A compartment, whereas in the ESLs, it was still in the A compartment, but associated with the nucleolus by published NAD data. And here, to get an understanding of how the gene was localized, we took the same samples that we produced for Immunogam, and we did cryofish to look at the gene localization. So here we put probes across the entire RBFOX1 gene, and then looked at it in ESLs and PGNs. And so you see the green foci here is the RBFOX1 gene, and the inside is showing it a bit more magnified. You can see in the ESLs it's quite compact, whereas in the PGNs, I hope you can appreciate that it's much more extended and decompacted. We measured the areas and saw that indeed there was a population of cells that had very high areas, sometimes even more than 1 micrometer squared. The other aspect, as I mentioned, we saw from the published NAD data that in ESLs, the gene was localized to the nucleolus. And so we measured proximity to the nucleolar periphery by labeling the nucleoli. And what we saw was indeed in agreement with the published data. Most of the cells in the ESLs had the gene at the nucleolar periphery, followed by the nuclear periphery, and then fewer in the nucleoplasm interior. And that was completely reversed in the PGNs. And now the gene was mostly localized in the nucleoplasm interior, with much fewer at the periphery and almost no cells at the nuclear periphery. The other aspect we wanted to look at was, as I mentioned, the melting we found was at the TSS of the gene. And so we wanted to understand if there was differences in the gene itself. So we put also probes specifically at the TSS at the middle of the gene, or at the TES, and different colors. And again, we performed cryofish. And what we saw in ESLs was that all 3 probes were mostly overlapping, and again, in this kind of compacted state, whereas in the PGNs, now the signal was much more spread out, and you could very easily visualize the different foci. We measured the distances between the probes and saw that in the PGNs, they had significantly higher distances between the TSS and the TDS compared to the ESLs. Again, sometimes reaching more than 1 micrometer in distance from one another. We saw some increase in the PGNs when we measured the distance to the middle of the gene, but it wasn't significant. And we saw no difference between ES cells and PGNs when measuring from the middle of the gene to the TES. So that was really exciting because it really well matched what we saw in the downgrade. So we then have this model where very large genes, again, genes that are longer than 400 kb that we measured here, can decondense or melt when they're very highly transcribed or very highly accessible. We see reorganization of their nuclear position, for example, RBFOX1 moving away from the nucleolar periphery. But we also saw, and I didn't have time to show you this, but I'm happy to show you this data later if you're interested. We also found that melting is generally related with a relocalization to the chromosome interior. So for the last part of my talk, I just want to talk about another interesting aspect of this work, and that was that we had two neuronal cell types here. So again, we had PGNs involved in memory and learning, and then we had dopamine neurons involved in reward and addiction. But they could have very strikingly different chromatin topologies. So here I'm showing a 5 megabase region where you see this kind of strong contacts in this large domain in the PGNs. They're not as strong in the DNs, not nearly. Or we have this stripe of contacts in the PGNs. Oops. The stripe of contacts in the PGNs at this gene dense region, which is depleted in the DNs. And so we wanted to ask, where are these differences strongest? And what does that mean for the regulatory information that's encoded there and for gene expression programs? So we first normalized the matrices and took a differential matrix between them. Here, more blue is stronger contacts in PGNs, and you see that stripe here, very strongly blue, and stronger contacts in DNs are in red. And then we looked at the distribution of these contacts and took the tails to ask where are the contacts most different, in this case looking at the 5% most differential contacts. And again, you see the stripe of contacts in PGNs. These blue contacts are very strongly differential between PGNs and DNs. And most of the contacts here were PGN specific, although you can make out some that are green, which are DN specific. So as I said, the goal was really to get at what regulatory information can be found in these contacts, what is describing them, and how is this related with gene expression. And so this was work with Lonnie Welch's lab, where at the base of these contact loops, we looked for ataxec peaks within each of the windows. And then within the ataxec peaks, we searched for differentially expressed transcription factor binding motifs. And just if I can guide your view here, look at this region that contains this stripe. Here we have the ataxec peaks from the PGNs, and then you see motifs for transcription factors that are very important for PGNs. For example, NeuroD1 and NeuroD2, developmental TS, but also involved in plasticity responses, for example, EGR1 as well. And you can see that there are very many of these transcription factor motifs. However, in the DNs, there's lots of ataxec peaks here, but there's almost no transcription factor motifs found within them, even ones that are highly expressed in DNs like FOXY1. So this was really exciting, especially when we looked at neighboring regions like next to this region where there aren't as many differential contacts or strong differential contacts, and you see those motifs in those cell types. Now, the other interesting aspect was when we looked at the base of these contacts, we found genes that were very either expressed or differentially expressed between the cell types, enriched in these contacts, and the ones that I'm highlighting here are all involved in synaptic plasticity responses. And indeed, when we looked at all of the contacts that were described by, for example, this NeuroD group of contacts, that NeuroD group of transcription factor motifs, we found a large amount of differentially expressed genes. Here we found 600, for example, of 1,800 total upregulated PGN genes. And when we looked at their gene ontology, again, we found genes related to synaptic plasticity, as well as a regulation of synaptic transmission. So if we go back now to our example, and we look at the region, this stripe region, which is contacting the downstream area here, when we zoom in and look at the contacts there, we actually find that they are contacting, there are contacts between these genes 3 or more megabases away that have these NeuroD group of transcription factor motifs in open chromatin regions. So we end up with this model where genes that are quite distal from one another can come together, and they're being brought together by accessible chromatin and possibly by the binding of TFs or the potential for binding of TFs. And that's quite exciting, especially when you think about responses to neuronal activation. And I'm thinking of the work by Jen Kremens, for example, showing new chromatin loops forming after neuronal activation. Just to say, we also found something similar in the DNs. I don't have time to show you today, but we also found these types of transcription factor motifs that describe genes that contact one another, in this case related to addiction pathways. And we think that these TFs are likely involved in the formation of interconnected hubs, which bring together these genes for these specialized functions. And it's quite reminiscent of polymer modeling studies that have predicted these types of specialized clusters. For example, from the Marendutzel lab, the self-assembly of specialized clusters coming together with strong binders, or from our own lab in Winick-Ng's lab, showing clusters that form when in the presence of a high concentration of bridging molecules. Don't really have time to show you this, but we found a lot of TAD border cell type specificity. 36% of borders were cell type specific when we did a multi-way analysis. And then I think I have a few minutes, so I just want to take one moment to just put our work into the context of what has been found previously, because it's really quite fascinating. There's this debate about whether gene activation coincides with or is preceded by chromatin contacts between genes and enhancers, genes and regulatory region. And I think, in very many of the 3D chromatin contact studies, you very clearly see the strengthening of contacts between genes and enhancers. But in other specific studies for specific genes, you might find other possibilities. For example, in imaging studies, like the PAX6 region, which when very highly transcribed moves away from its enhancer, or in the SHH locus, which when it's transcribed highly in NPCs can move away from one of its enhancers as well in spatial proximity. And we find evidence for both the strengthening of contact and the increase in spatial separation. For example, we find strengthening between contacts when you have transcriptional activation, like the pcbh example of the contacts to the FGF1 gene. We also find that you can have gain of contacts depending on the genomic neighborhood, as well as the regulatory information that's encoded in those contacts. And then we find the opposite. We find that gene activation can be accompanied by large-scale decondensation events and actually increasing in spatial distance. And that's where I think this is really impactful, because these types of events have been shown previously in very specific conditions, but we're the first to be able to identify them unbiasedly and in complex tissues or in very highly specialized cells. So I think overall our study is really about these fascinating differences between how 3D architecture relates with or impacts on gene expression. And it really does suggest that it's highly specific of the locus, of the cell type, of the neighborhood, and as well as the context. And excitingly, moving forward, we can look at how it impacts on physiological states. And so just in summary, I showed that GAM is well suited for applications in situ and complex tissues. And it can be done in relatively low cell numbers. All the maps I showed you today were with about 1,000 cells. I showed that genome architecture is highly cell type specific in brain cells and at multiple scales, and that those folding patterns reflect cell specialization, especially, for example, at the loss of organization or melting of highly transcribed genes when they're very highly expressed or with high accessible chromatin. Also, between specific contacts between differentially expressed genes, which contain these complex networks of transcription factor motifs. So with that, I'll just say thank you very much, especially to my co-first authors, Isabella and Alexander, as well as Luna, who did a lot of the RNA-seq and ATAC-seq analysis, as well as producing ATAC-seq data, and Dominic, who developed the Neutron pipeline, as well as everyone else in the lab who's involved, and our collaborators, especially the Lonnie Welch lab for the TF analysis, and Mary McDemmy's lab for the polymer modeling study. So thank you very much, and happy to take questions.